Your career begins here at Satyabama Institute of Science and Technology, a deemed to be university, a nationally recognized and globally acclaimed destination to accomplish your academic aspirations. The Colonel Dr. JPR Research Park, along with the International Research Center, houses advanced research facilities that have 13 centers of excellence, 10 plus technology transfers, 100 plus patents, 10,000 plus reputed publications. The Satyabama Center for Advanced Studies is a state-of-the-art facility built with the aim to cater to the futuristic aspirations of our students with the aid of a metaverse studio, drone labs, makers space, media center, advanced characterization facilities, ICT-enabled classrooms, and supercomputing laboratories. The engineering departments are equipped with sophisticated infrastructure, fulfilling the academic and hands-on requirements of our students. The architecture department is furnished with sophisticated design studios and spacious drawing halls, all of which support the learning process of our postmodern generation architects. The Satyabama General Hospital, along with the School of Dental, Nursing, Pharmacy and Physiotherapy, have advanced clinical facilities. The monumental Central Library houses a diverse collection of books, periodicals and journals. Additionally, it also has an open-air theatre with a seating capacity of 1,000. The Management, Arts, Science, Law and Hotel Management departments comprise of all the necessary and required facilities, creating the perfect ambience for our far-sighted students and catering to their skill set needs with the end goal of equipping them to live their dream. Our chain of laboratories with practical exposure equips our students to build a successful career. Our institution has a team of dynamic and outstanding teachers who are ever reachable to our enthusiastic students, promoting the spaces to learn and places to grow. Greenery around the campus greets and accommodates our students, who are the backbone of a developed nation, edging them on to achieve the summits of their success. Satyabama Institute of Science and Technology.
Good morning, everyone. A very warm welcome to all the participants to another session of Ocean Research Knowledge Series 2026, organized by the Center for Ocean Research and National Facility for Coastal and Marine Research, Satyabama Institute of Science and Technology, with the support of Ministry of Earth Sciences, Government of India. Today's expert lecture is on tiny fish, tremendous insights, zebrafish as a biosentinels in modern environmental toxicology. We are pleased to have with us Ms. Soumya Prasad, Scientific Assistant, Center for Ocean Research, Satyabama Institute of Science and Technology, who will be delivering today's lecture. Ma'am, a very warm welcome to you. I now invite you to take over the session. Over to you, ma'am. Thank you.
Thank you, Sumesh. I'll just share my screen. Give me a moment. I hope my screen is visible.
Yes, ma'am. Yes, ma'am.
Right. So before we begin today's talk, let me introduce myself. I'm Soumya Prasad, Scientific Assistant at Center for Ocean Research, Satyabama Institute of Science and Technology. Before we begin today's talk, I would just like everybody to imagine collecting a water sample from a polluted coastal environment. The chemical analysis of that particular sample will tell us the contaminants that are present, right? But can chemistry alone tell you whether these contaminants are biologically active, or are they going to be affecting the aquatic life or causing cellular damage, or whether they are capable of altering the development and the behavior of the organisms? Now, this is where the environmental toxicology comes into purview, where this tiny fish can give us tremendous insights. So today's talk will be about zebrafish as biosentinels in modern environmental toxicology as a part of Ocean Research Knowledge Series. We'll just begin with a quick introduction. Zebrafish are small tropical freshwater fishes from the minnow family, which is the Cyprinidae family, native to the Northeast India and Pakistan. They are well known as a common aquarium fish, retailed under the name of zebra danio. There are a variety of zebra danios available in the aquarium fish markets. And it's a molded vertebrate organism used in laboratories to help scientists investigate the biological processes. It's a small fish, about 1 to 1.5 inches. Now, if you can see here, historic and most recent sites where zebrafish have been reported in India, Nepal, Bangladesh, and possibly even in Myanmar. And you can see some pictures of fishermen collecting wild zebrafish in the rivers of Meghalaya. And coming to the taxonomical classification of the zebrafish, it belongs to the kingdom Animalia and the family Cyprinidae. It belongs to the phylum Chordata and the subphylum Vertebrata. The genus and the species is Danio, which is commonly used in laboratory toxicological studies. So coming to the biological features of this fish, it has unchanging 5 pigmented horizontal blue bands on the side of the body. The stripes spread from the end of the head to the end of the caudal fin. So the growth of zebrafish happens up to 6.4 centimeters, that is 2.5 inches. It is an omnivorous feeder, feeding both on plant and animal matter, and they have a lifespan of 1 to 2 years. Now, why this zebrafish is very important is because they show sexual dimorphism. So the males are slender, and the body has a pink or yellow tinge with golden stripes present in between the blue stripes. And females tend to be less pinker than males. They have a silvery lining on their body, and they look fatter and they're streamlined. You can see a bulge at the bottom of the animal. You can see that's where the ovaries are housed, and they carry the eggs in their belly. So these female fish tend to have whitish belly, and silver stripes are present amid the blue stripes instead of golden color in the males. So coming to the life cycle of the zebrafish, you can see the time to fertilization is 0 to 24 hours. So the entire developmental cycle of zebrafish from a single-celled embryo, single-celled fertilized embryo, to a hatching larvae fry stage, it comes in 24 hours. So these are the different lifestyle stages of zebrafish, where you can observe the 6 somite, 14 somite, prime 6, and the protruding mouth stages very clearly under the microscope. So here are some images captured with the microscope. So that's the early embryonic development stages of zebrafish, where you can see the 3 somite stage, 90% epivolley, 18% 18 somite stage at the 18-hour cycle and all that. Coming to the history of zebrafish, I think this is very well known. The use of this zebrafish as a model organism was pioneered at the University of Oregon by George Streisinger. He's also known as the founder father of zebrafish developmental and genetic research. And in 1990, Christian Wolhard, she was the one who conducted large-scale mutagenesis of zebrafish in her laboratory, and she is associated with Oxford University in UK. And Thomas Look, he was the scientist who used translucent zebrafish embryos to study cancer genetics. This study began in 1995. Why we use zebrafish as a model organism is because the zebrafish has a very short reproductive cycle. The genome is already fully sequenced, and embryos are transparent. The case of maintenance of large numbers in a small laboratory scale machine is also possible. It doesn't take up much space. Adult zebrafish are able to regenerate their organs. Simple genetic manipulation techniques can give you transgenic models of zebrafish, and over 70% of the annotated human genes have true orthologs in zebrafish genome. That is, they are very similar to our human genome. And high number of offsprings can be gotten in a very short period of time, and the development is external and very, very, very rapid. So this is why zebrafish is used as a model organism. They have optically translucent embryos. Rapid hatching of eggs happen within 24 hours. You can see the larvae also. And the maintenance cost is significantly lower than those for mammals. For mammals, you will have to house separately. You will have to give them proper feed. Zebrafish feed and the housing requirements are very small, and it can be done in a very small laboratory space. Zebrafish genome is very amenable for molecular and genetic analysis, and they are fertilized, and as they develop outside the mother's body, it is an ideal model organism to study early embryonic development. Coming to the genetic information of the zebrafish, over 26,000 protein coding genes are present. The chromosome complement is 2N, that's 50 chromosomes. 2 metacentrics are there at 6 and 11, and 8 submetacentric chromosomes are there. And remaining 15 chromosomes are subtelocentric. And total of 26,206 protein coding genes are there. And it demonstrates more than 75% similarity to human genes. And over the half of chromosome-rich pairs have GC-rich sequences, and these are found at the pericentromeric locations. So in this zebrafish genome, as I said already, 70% of protein coding human genes are related to genes found in zebrafish, out of which 84% of the genes are known to be associated with human disease. They have a zebrafish analog, the counterpart is there. They can be cloned very easily from somatic cells. They can be made transgenic. And as a vertebrate, the zebrafish has the same major organs and tissues as humans. Their muscle, blood, kidney, and eyes share many features with human systems. So studies in zebrafish models can be extrapolated to human models also. So when you come to zebrafish as a disease model, it can be used as two models, one for disease and one for toxicity studies. Coming to the disease, in the clinical toxicology purview, we have zebrafish models for cancer, cardiovascular diseases, neurological and behavioral diseases, genetic disorders, metabolic disorders, infectious diseases, and immune disorders can be studied. Coming to the environmental toxicity studies purview, we can also study the developmental toxicity, neurotoxicity, cardiotoxicity, hepatic and nephrotoxicity, the reproductive toxicity, and the genotoxicity using the zebrafish model system. So we'll be studying in detail about these kind of endpoints which can be done using the toxicity assessment. Zebrafish mainly is used in ecotoxicological studies due to its simple and inexpensive maintenance. And this is a good alternative to screen the endocrine disrupting properties and toxicity of various natural and synthetic compounds that are present in the environment as a pollutant. So coming to the practical applications of zebrafish in toxicity assessment, You can see that developmental toxicity studies can be used to assess the impact of toxins on embryonic development, monitoring the morphological changes and organ formation, behavioral toxicities to study the effects of toxins on zebrafish behavior by assessing the locomotor activity and the cognitive functions. The C-tox assessment, that is the cardiotoxicity assessment, evaluates the impact of toxins on heart development and function. And you can monitor the cardiac activity in real time because the embryos are transparent and you can very clearly see the beating heart under the microscope. And these are also used in PK and PD studies, drug metabolism and biotransformation studies. So they possess a functional liver-like organ, the yolk sac, during early embryonic development. So this is useful for studying the drug metabolism and biotransformation inside the body. So this also comes handy in the point of ecological toxicity, where biotransformation of contaminants can happen inside the in vivo system. And high throughput screening is possible. And for environmental toxicology, we particularly use this model to assess the impact of pollutants and contaminants on aquatic systems and to understand the broader ecological implications of the toxins that are present in the system. So here, a single zebrafish, you can do the toxicity effects, the effects of pesticides, the effect of chemicals that are run off from the water systems into the aquatic food chain. It goes and you can just study the developmental toxicology, the endocrine disrupting, the reproductive toxicity, neuro, immuno, and genotoxicity. And you can also couple this studies by studying the intrinsic mechanisms using omics technologies like transcriptomics, metabolomics, and microbiomics, where you can understand how these chemicals affect the upregulation and downregulation of certain genes, the expression of certain proteins. And coming to the microbiomics part, the dysbiosis of the host microbiome that is there in the zebrafish. And if you can look at the feeding cycle, the life cycle of the zebrafish, you can see that the 0 to 72 hours post-fertilization stage is known as the embryonic stage. And the early larvae stage occurs from 72 hours to 13 days post-fertilization. The mid-larvae stage from the 14th day to the 29th day post-fertilization. And you can very clearly see the juvenile stage developing from the 30th day to the 4th month after fertilization. This juvenile stage is very optimal for doing the toxicity analysis. And sexually mature adults, when they become sexually mature at around 6 months after fertilization. So coming to their feeding cycle, up to the 13th day, up till the early embryo stage, you will be need to do a continuous feeding regimen for the zebrafish. And up to the 60th stage, you can do it 3 to 5 times daily, which is recommended optimally. And from the 60th day, you can give 1 to 3 times proteinaceous feed daily. These are some maintenance parameters for zebrafishes which are recommended. The room temperature of maintenance of the zebrafish system should be around 28 to 29 degrees Celsius. The relative humidity should be maintained around 70%, and the light intensity should be maintained around 300 lux. Photo period is very important for zebrafish maintenance. So 14-hour photo period and 10-hour dark incubation is mandatory because this aligns with their circadian rhythm and mimics their natural environment. So this maintenance is very important. Coming to the noise and vibrations, it is advised that it is kept free or minimal if possible. So when you are trying to culture them in the lab, you can use vibration-free magnetic motors. So zebrafish is very sensitive to noise, and keeping the lab noise-free or vibration-free as much as possible is recommended for maintenance of zebrafish. Now, coming to the fish toxicity testing, we have some key OECD guidelines for the fish toxicity testing, which will be going in detail. So the fish OECD 203, you can see here. Yeah. So this is adopted by the OECD on 18 June 2019, and it is a semi-static or a static flow-through condition. This assay can be conducted. So 5 different concentrations minimally has to be taken in a geometric series, the concentration of your pollutant. And the fish should be juvenile and should be originated from the same source and population to ensure uniformity in the assay. And they should be of the same age and have the normal appearance. So at the endpoints of this assay could be mortality, abnormal appearance, and behaviors. Now, coming to the OECD toxicological guidelines, you should understand how to perform the test and how when your test is considered valid. So validity of the tests, you need to ensure that the control, the mortality should not exceed more than 10%. The dissolved oxygen concentration in the static, semi-static conditions should always be greater than 60% of the air saturation value in all test vessels throughout the exposure time period. The analytical measurements of your test concentrations prior to application to the well plates in the lab is necessary. So here you can see the effect of hydroxychloroquinone on zebrafish embryos, different concentrations, and the survival is given. So as the concentration increases, you can see very clearly the survival rate decreases, and there is also a very clear evidence of malformations that are formed in the embryo. And this is the short-term embryo toxicity test, OECD test number 212. So here you can see that this focuses on chemical effects during the early vulnerable stages of zebrafish development. So this test you have to begin within 30 minutes after the eggs have been fertilized. So you can screen for mortality, malformations, and behavioral changes in the zebrafish. Here also the validity parameters matter. The validity should be greater than 60% of the air saturation value, the dissolved oxygen, and the water temperature in the controls and the test wells should not be greater than 1.5 degrees Celsius. That is, when you're adding chemicals that are exothermic in nature, you have to understand and make sure that the water temperature in the test wells does not go beyond or lesser than 1.5 degrees Celsius. Analytical test measure is compulsory, and you can also imply to have relevant solvent controls if you are dissolving or dispersing your drug in a particular solvent. The fish embryo acute toxicity test, OECD 236, is very commonly accepted and done in most of the labs. So this test is specific to zebrafish embryos. So first you will breed the zebrafish by spawning them in a particular ratio that is optimized in your lab. Generally, we take one male is to one female in our lab, which we do. And after collection of the embryos, you can just screen the embryos for normal development. And once your embryos are screened and the dead embryos are removed, you can distribute them in the well plates and add your chemicals according to the test concentrations. So this test replaces the need for testing on adult fish, and the endpoints are mortality, abnormal development in the tail, and pericardial edema can be very clearly observed. So endpoints could be coagulation of the fertilized eggs, lack of somite formation, lack of detachment of the tail bud from the yolk sac, or delay in the formation of somites, and lack of heartbeat. So here, the certain parameters for validity of this test are very important. So the fertilization rate of all the collected eggs should be greater than or equal to 70% in all the batches tested. The water temperature should not vary very greatly. It should be around 26 plus or minus 1 degree Celsius. Overall survival in the negative control, that is the untreated control, should be greater than 90%. That is when your test is considered valid. So if there is mortality in the control, then you will have to consider redoing the test. And the exposure to the positive controls should result in minimum mortality of at least 30 to 50%. So if it is less than 30% mortality, then you should consider increasing the positive control concentration or redoing your test. And the hatching rate in the negative or the untreated control should be greater than 80%. And the dissolved oxygen in the negative and the highest test concentration should be maintained at 80% of air saturation level. And a couple of other tests are there, the juvenile growth test, the fish short-term reproduction assay, and fish sexual development test. So this particularly is very significant in ecotoxicology. because this assesses the chemical effects of endocrine disrupting chemicals on sexual differentiation and development of zebrafish by identifying potential disruptions and alterations in sexual differentiation. You can conduct this test in the embryo stage throughout the sexual differentiation period, that is 60 days post-fertilization. The endpoints is a very important biological marker, which is vitellogenin. The sex ratio can be determined, and the histopathological analysis of the gonads can be done to understand what is happening inside the tissue. Here, if you can see, the vitellogenin protein, which is an important biomarker, is a female-specific egg yolk precursor protein. This male fish possesses the gene and liver receptors to make the vitellogenin, but in males, this gene remains silent. When exposed to endocrine disrupting chemicals, this gene gets activated in the male fish and vitellogenin is expressed in the male fish. So this finding of this vitellogenin in male fishes is a very clear indicator of pollution by estrogenic endocrine disrupting chemicals in the ecosystem. So this analysis can be done by taking the blood plasma or non-invasive tests by collecting the fish mucus from the skin. You can understand the expression of this vitellogenin. And the EC assay, which is detection of the endocrine active substances acting through estrogen receptors. So here you can see a transgenic zebrafish line that is maintained. Here it is tagged with a GFP, and you can very clearly understand this estrogen receptor localization where the particular toxicant is going and acting. So endpoints assessed could be the estrogenic activity, the localization of response, thereby giving you a quantitative data of how much impact has happened. And coming to the OECD test 305, bioaccumulation in aqueous and sediment phases. So here you can study the bioaccumulation potential of chemicals like heavy metals, your endocrine disrupting chemicals, and your other potential hazards in the environment. And it also helps you understand the environmental fate of chemicals, whether these chemicals can be transferred from the aqueous phase or the sediment phase into the fish. You can determine the bioconcentration factor, and you can do the heavy metal analysis in the tissue by doing an ICP OES, ICP-MS, or AAS, and sediment interaction analysis also can be done. So benefits of using zebrafish in toxicological assays is that high throughput screening. You can screen a large number of embryos in smaller volumes, and environmentally relevant concentrations can be exposed in the lab, and they are a very good alternative to mammals. So they reduce ethical concerns in toxicity testing. So they are ideal for early life stage testings, and availability of transgenic lines and CRISPR technology to study mechanistic pathways is a very big advantage of using zebrafish in toxicological studies. Coming to the emerging trends in zebrafish-based toxicology, the 3R principle is widely adopted by using zebrafish as an alternative model. That is refinement of the concentrations, reduction in the number of embryos, the number of organisms, and the replacement of animal testing, and integration of omics, that is genomics, proteomics, and metabolomics can give you very great mechanistic insights on how a toxicant acts on the animal. And the behavioral toxicity can be studied using automated tracking systems, which are available now, analyzing locomotion and neurobehavior, and their social behavior, whether they are schooling or they are becoming aggressive inside the tank or they are isolating themselves. All these parameters can be studied by automatic tracking systems. Now coming to the mortality endpoints, that is, you can very clearly see the embryo malformations here. The first image is a normal 96 hours post-fertilization zebrafish. You can see a very clear swim bladder, inflated swim bladder. And in the second image, you can see the localization of red color as indicated in the arrow mark. That is a representative of heart malformation in a 96-hour HPF larva. And the third image is a representative image of a coagulated embryo, which is the mortality endpoint of the embryo toxicity assay. And when you come to the fourth and the fifth image, you can very clearly see uninflated swim bladder, pericardial edema, and yolk sac edema. So these malformations are indications of toxicity in the system. And a couple of other malformations like the end tail malformation or the short tail formation or complete scoliosis, that is complete curvature of the vertebral column. All this you can observe very clearly under the microscope. In our lab, we have done acute toxicity testing of selenium nanomaterial exposed to zebrafish. Here, we have exposed these zebrafish embryos to increasing concentration of chitosan selenium nanomaterials. And you can very clearly see that the number of viable embryos start decreasing as the concentration increases. And coming to the heart rate, as the concentration increases compared to the control, the number of heartbeats per minute, that is the cardiac rhythm, is affected. And you can very clearly observe bradycardia, that is lowering of your heart rate in increasing concentrations. Now, very clearly, we can see more than 250 micrograms per ml concentration. There is a clear mortality observed. And hatching rate is also greatly reduced as the concentration is increasing. Very minute concentrations, that is even 15.625 micrograms per ml, has a very significant impact on the hatching rate as well as the viability of the embryo. You can very clearly see the stereo images of embryos where you can see the malformations and the developmental anomalies. The first image is the control image. The second image is treated with a positive control. You can very clearly observe the complete crystallization of yolk sac protein and coagulation of the embryo in the positive control. Similar effects can be observed in concentrations image G, H, and I with 250 micrograms per ml, 500 micrograms per ml, and 1000 micrograms per ml concentration of selenium nanomaterials. And you can very clearly see the developmental delay compared to the normal control untreated embryo. You can see pericardial edema and yolk sac edema, and developmental anomalies are observed. Why this is very important is because these embryos also show decreased heart rate, so their cardiac functions are also impacted. Now, clearly, these selenium nanomaterials nowadays people use in a lot of aquaculture feeds to kind of, remove or control the pathogenic microbial growth inside the fish gut. So these are mixed with aquaculture feeds to reduce the spread of diseases. But very clearly, you can see that these selenium nanomaterials have toxic effect on the developing embryos. And we have also done the toxicity profiling in the larval stage of the embryo. Even in the larval stage, we could observe decrease in viable larvae. And the bradycardia was observed in treatment concentrations as they increased. Significant bradycardia was observed. And very clearly, you can see the malformations, uninflated swim bladder, enlarged eye, that is macrophthalmia, bent tail or end tail malformations, and membrane distortions, which are indicated in the black arrow, with the black arrows. And coming to the animal study protocol that we have done in our lab, we have done the toxicity study using water dilution method. We have divided the animals into 7 groups, and we have used a juvenile zebrafish with mixture of male and female animals. And each group we had 7 animals with increasing concentration of nanomaterial, selenium nanomaterial treatment. The group 1 was the control group, and the group 2 was treated with positive control. And you could very clearly see in the histopathology of the gill, gut, and the hepatocytes in the control group without selenium nanomaterial exposure, we could very clearly observe intact structural morphology of the gill, the gut, the intestinal villi, the mucosa, the goblet cells. And when you come to the liver cells, that is the hepatic tissues, intact and liver parenchyma is observed. Coming to the positive control, you could very clearly observe the degradation of primary and secondary lamellae, and you could very clearly observe the diffuse villi atrophy, that is the complete villus morphology is changed, and you could see mucosal necrosis that is observed, which is indicated with small blue arrows in the image. And if you could observe in the hepatocytes, you could see severe vacuolar degeneration and unidentification of sinusoidal spaces. Now, as the treatment concentrations increased, the selenium nanoparticle treatment concentration increased, the highest concentrations, if you could see the 300 mg per liter, 400 mg per liter, and 500 mg per liter, the complete gill morphology is damaged. The primary and secondary lamellae are degenerated and show gill atrophy. And in the gut, you could very clearly see mucosal necrosis, which are very diffuse. And when it comes to the hepatic cells, you can see degeneration and degeneration of the vacuoles and unidentification of the sinusoidal spaces in various degrees very clearly. And so prolonged exposure to this selenium nanomaterial could cause serious implications in the gut lining and the gut bacteria of the zebrafish, thus affecting the overall health of the animal. So these visceral anomalies are completely ignored when it comes to pathogen control in aquaculture farms. So I believe that our study could, bring about safe exposure concentrations to the animals. So majorly the gut dysbiosis is a phenomena that affects the animal because the gut-brain axis is there, and this also contributes to the overall health implication in the animal. And yeah, so coming to the zebrafish life cycle, you can the toxicity studies can be summed up using this image very clearly. The OECD 203, 236, 212, 215, and 210 can be done from the embryo to the larval stage, and the 229, 305, and 234, the OECD assays, could be done from 21 days to the sexual maturity stage. So each stage of the zebrafish can help you understand, how your contaminant works in the environment and how it affects the aquatic life. So coming to our closing statement. So the embryo, because it's a rapidly developing system, it has optical clarity, genetic malleability, and external development. It is very useful in toxicological testing. Larva also has optical clarity and genetic malleability, and it can help you in high throughput sequencing. The adult has a vertebrate neural structure. The maintenance is low cost and very easy to maintain. It requires a very small space. The genome of the adult zebrafish comprises of several orthologs to human genes. And this zebrafish, they serve as an invaluable model for toxicity studies, offering unique combination of genetic similarity and relevance to human health, the cost effectiveness in research, and practical applications across various toxicological assessments. So thank you for your patient listening. I'll be happy to take questions.
Thank you so much, ma'am, for that informative presentation. So participants, if you have any doubts, please ask in the chat box. So one question is there, ma'am.
Yeah.
What is the meaning of transgenic fish, and is there foreign gene introduction in zebrafish?
Okay, so this transgenic zebrafish is particularly when you knock out certain genes in a zebrafish by altering its genome to mimic certain disease models. That is transgenic zebrafish. So various transgenic lines are available for cancer studies, cardiovascular disease studies, and neurotoxicity studies for Alzheimer's and Parkinson's and all this. So yeah, that's about it.
Okay, there is no other question, I believe. So thank you, ma'am, for answering the questions, and thank you all for all the participants for your active participation. Before we conclude, I would also like to inform everyone that I have shared some useful information in the chat box, including consultancy services and related details from COR and NFCMR, MSc Marine Biology related admission related details, and also internship opportunities being conducted at COR and NFCMR during August month. And I request interested participants and students to kindly check in the chat box and make use of the opportunities. And with this, we conclude today's ORKS 2026 session. On behalf of COR and NFCMR, I sincerely thank Ms. Soumya Prasad, ma'am, for the excellent session, and all the participants for joining us today. Thank you, everyone. Have a wonderful day.
Thank you, Sumesh.